Dear student, welcome to the Medical Genetic course. Today's lecture is DNA repair mechanism. By the end of this video, you are expected to identify the cause of DNA damage and the types of DNA repair mechanism. You also expected to give examples of disorders caused by defect in these mechanisms. The content of this lecture include the cause and types of DNA damage, the type of DNA repair mechanism, and disorders caused by defect in DNA repair mechanism. How does DNA damage occur? Naturally occurring mutations are referred to as spontaneous mutation and are thought to arise through errors in chromosomal division, DNA replication, or in the cell throughout reactive oxygen species. Environmental agents that cause mutation are known as mutagens that include natural or artificial ionization, radiation, and chemicals, or physical mutagens. It has been well established that an accumulation of mutations in DNA, whether caused by external source such as ionization, radiation, gamma rays, X-ray, ultraviolet ray, chemicals, or internal source such as reactive oxygen species, has the potential to cause a cell to undergo carcinogenesis and increase the risk of the development of cancer. DNA damage results in deletion, mutation, cross-linking with protein or other bases, which can affect function of the protein. In normal cells, the occurrence of mutation in DNA, if left unrepaired, would have serious consequences for both the individual and subsequent generation. The stability of DNA is dependent upon continuous DNA repair by a number of different mechanisms. Genomic integrity is protected by the DNA damage response, which is a collection of mechanisms that sense DNA damage, signal its presence, and promote repair mechanism for different types of DNA damage. When there is DNA damage or replication stress, DNA damage response activate a collection of DNA repair, which we will talk about in the next few slides. If the cell can't repair itself, then it goes through apoptosis. One of the most important mechanisms that cells go throughout is checkpoints during cell cycle. There are several crucial protein and enzymes that works as checkpoints and make sure that DNA is not damaged before it goes to cell division. So, a question is, what happens if these pathways are mutated or it doesn't respond? Unfortunately, it will promote genomic instability and predispose individual to cancer. People who are born with inherited mutation of DNA repair genes fail to repair their DNA damage effectively and have great risk of developing cancer. They are known to be suffering from genomic instability syndromes. So, DNA repair includes a wide range of processes. First of all, cells have to identify the type of DNA damage in order to introduce the correction that restores DNA to its original structure. If the damage is in the single strand, then there are three types of repairs. The first one is mismatch repair. The second one is nucleotide excision repair. The third one is base excision repair. If the damage is in the double strand, then there are two types of repairs. The first one is homologous recombination. The second one is non-homologous end joining. The first single strand repair mechanism is mismatch repair mechanism. This process corrects mismatch bases introduced during DNA replication. Some errors are not corrected during replication, but are instead corrected after replication is completed. This type of repair is known as a mismatch repair. which is responsible for maintaining genomic instability. In mismatch repair, the missed errors are later corrected by the enzyme that recognize the errors as emerging loops where the two strands don't precisely align. Cut at both ends of the error by endonuclease, excise the mismatch base, polymerase the new correct base, and seal the gaps by ligase, as we see in the picture. Mutation in the mismatch repair mechanism cause hereditary non-polyposis colorectal cancer, or Lynch syndrome. The second type of single-strand repair is nucleotide excision repair. In humans, UV radiation from the sun is the leading cause to skin cancer. UV exposure or radiation induce lesion and form an extra covalent bond between adjacent same strand pyrimidines, particularly thiamine. The linked thiamines are called thiamine dimers, which cause alteration in the structure of DNA and inhibit polymerase and arrest replication. Nucleotide excision repair, the protein produced from these genes called NER, which play a variety of roles in this process. They recognize DNA damage. This repair mechanism is used to replace region of damaged DNA up to 30 bases in length. The third type of single-strand repair is base excision repair. It's responsible for correction of small base, 1 to 5 nucleotide lesion. Specifically, corrects error that result from oxidative damage. The damage repaired by base excision repair is the result of oxygen-free radical produced from chemical reactions such as metabolism and transcription. An example of defects in nucleotide excision repair is Ectoderma pigmentosa. It's a germline mutation in XPA gene, which is involved in repairing damaged DNA. XPA, this disease is an autosomal recessive that reflects defect of nucleotide excision repair or deficient DNA polymerase, both of which allow thiamine dimer to stay and block replication. Also, mutation in ERCC excision repair A gene caused Cockayne syndrome. It caused microcephaly and growth failure, skeletal abnormality, mental retardation, and other symptoms, as shown in the picture. This summarizes how DNA is repaired when the damage is one in one strand. Excision and mismatch repair differ in cause and error. UV-induced pyrimidine dimer versus replication errors, and in the type of enzyme involved. The other types of DNA repair is double-strand DNA damage, which is much harder to repair because the damaged helix has no template from which to copy and replace the affected bases and nucleotide. There are two major pathways that can repair double-strand breaks: homologous recombination and non-homologous enjoining. Usually, double-strand breaks is induced by exogenous agents such as ionizing radiation, or it occurs spontaneously during cellular processes. The impairment in these pathways is associated with defects and pathologies including genomic instability, immunodeficiency, and cancer. Homologous recombination is a repair mechanism used for a double-strand break. This process is highly complex. It requires strand exchange and alternate base pairing and complex structure and multiple proteins. It uses the homologous sister chromatid or chromosomes that has similar DNA sequence to the damaged strand. The identical sequence serve as the template for repair and enzyme. However, the non-homologous end joining is often leads to permanent change in the DNA sequence. Because this process is error-prone repair mechanism. It does not rely on a template. It connects the break end without a homologous template. It uses a short DNA sequence called microhomologous. It's not accurate, but protects the genomic integrity from possible chromosomal translocation that can occur through homologous recombination. Defects in double strand break can have severe consequences. For example, mutation in BRCA1 and BRCA2 genes are associated with breast and ovarian cancer. BRCA1 and BRCA2 mutation leads to drastically increased chromosomal instability. Both gene products have been shown to be related to double-stranded break. Also, autosomal recessive mutation in ATM gene cause ataxia telangiectasia. ATM gene involved in DNA repair and control cell division. It results in a defect in a kinase that function as a cell cycle checkpoint. In this disease, cells proceed through the cell cycle without pausing after replication to check the new DNA and to repair any mispaired bases. In summary, we have talked about the causes and types of DNA damage, the DNA damage responses, and mechanism how to deal with DNA damage.